everyone. Hi, everybody. We're going to get started here soon. So thank you all so much for attending. I'm absolutely astonished by this amazing crowd. Thank you so much all for joining us. My name is Marissa Zuckerman. I'm a communications associate here at the Boyce Thompson Institute. I've also been responsible for coordinating this year's Art at BTI event. I'm very pleased to welcome you to this evening's Art at BTI reception. Now in its 5th year, Art at BTI continues to celebrate the collision of art and science at BTI and beyond. This year's exhibit, Juxtaposition of Light and Texture, features the floral and architectural photography of Nancy V. Ridenour. We'd like to thank Nancy for allowing us to display her work at the Institute over the past few months. Her work has been a joy to everyone who has stepped foot through BTI. We would also like to thank Cayuga Landscape for their generous support of this evening's event. We are grateful for Cayuga Landscape's continued support of events and education and outreach programs. Thank you as well to Katie Walker and Agava for donating a portion of tonight's wine and catering services. Lastly, we would like to thank our generous donors who make events like Art at BTI possible. We hope you will consider making a gift, which will enable BTI to continue to host events such as this. To start this evening's presentations, we will give a brief video to introduce you to the Boyce Thompson Institute, our research, and our mission to advance and communicate scientific discovery. Thank you. Can I just mic you? So I'm now pleased to present to you President and CEO of the Boyce Thompson Institute, David Stern.
I think that video shows you how we all got hooked on science. It's just so cool. If it was only that cool every day. It is at BTI, it's just other places. It isn't. I want to call out Marissa. As she said, she coordinated this event. It's a big deal. A lot of people pulled a lot of weight, Keith and Stephanie and all their staff, but Marissa, you were the centerpiece, and she's going off to school, so we're really proud of her. We're going to miss you, Marissa, but thank you for pulling this off. So please, let's give Marissa a hand. good people behind the scenes can make anyone look like a genius. You can take that home. I do want to thank you all for coming. If we could get this great of an attendance for our scientific seminars, that would be awesome. I think everybody in Ithaca is in the room except my son, who's out there somewhere wandering the stairways. It's really, more seriously, that's true, but to have this kind of engagement with the community is really important. I think it's, we've finally realized we have to stop saying that BTI is Ithaca's best kept secret. It's not a secret anymore. You've all shared it, and you've shared it with people, other friends, and you've shared it on social media and lots of other places. So, so thank you for being part of our family and for the contributions that have made this event possible. So I think you've seen a good idea of what BTI is. I just came back a couple of weeks ago from a special trip. I went to Butte, Montana, where William Boyce Thompson went to school until they told him to go do something better than living in Montana. I don't think Butte's changed that much since the 1860s, maybe a little bit. The uptown part really looks like a frontier town. And I was there to present a scholarship that a donor had given in his name, and the donor couldn't be there, and I was delegated to go, and I got to see where he grew up. And when you compare Montana to what we do here now, I think he would be proud. So, so BTI is about discovery. William Boyce Thompson discovered himself and a lot of things during his life. We're trying to discover secrets of life and put them to use where we can. And we also build careers here. So part of our career building is science communication. Part of it is the science itself. Part of it is just getting the public interested and excited and understanding what does plant biology mean besides farming. It's not just farming. It's really intricate, fascinating science. And I know Nancy's going to tell us why it fascinates her. So as was mentioned by Marissa, this is our 5th Art at BTI. So I've been president for about 13 years. So it was quite a bit of time into my presidency when somebody came up with the idea of engaging local artists and realizing that the beauty in art and the beauty in science are in fact related. And it could be said, in fact, that there's science in art and art in science. And certainly there's art in science because some people come into a laboratory and magic happens. I'm not sure if I was one of those people. Joyce is one of those. She's people literally call her a magician. So there's art in what you do. It's that sixth sense. It's feeling the plant. It's feeling the life. It's feeling the biology. And of course, in art, if you weren't inspired and emotional about it, but also scientific, that the lighting is right and all that, and we're going to hear about that from Nancy. So I think the connection between art and science is an ancient one, and it's one that we have learned to appreciate in a public way at BTI. Plus, when I came here and you walked around the halls, it was boring. It was pictures of, people who looked a little bit like me, and that's not very interesting. We can do better, and we have done better. So this is something that we look forward to continuing for the indefinite future, as long as there are artists willing to share their work as generously as Nancy has. So the featured artist tonight, as we've heard, is Nancy. She's a digital photographer, as you've seen. So she likes taking pictures of the natural world, and that's a wonderful thing, including flowers, birds, landscapes, and so on. And she's going to tell us about her inspiration, her methods, and so on, so you can understand a little bit of what's behind the work. I often go around and I say, wow, what a pretty picture, but I don't really think about how hard it must have been. So I look at my phone and I see all the blurry images of my supposedly beautiful orchard, and so there's a real science to it in an art, and I look forward to hearing about your inspiration. And in art at BTI, we always pair, it's like a wine pairing. If you think about it, we pair a scientist and an artist. So the scientist, Joyce Van Eck, you'll meet in a few minutes. So she's an associate professor at BTI. She's homegrown. She's been here for a while, and she, as I said, is a magician with genetic manipulation of plants. And what I mean by that is using genetics to figure out how plants grow and develop, and to improve nutritional qualities of plants, and so on. And she has this really interesting project right now on something that has 1,000 names. Like a banana is a banana, but ground cherry, goldenberry, golden cherry, pichu berry, they're all the same thing. Husk cherry. How many of you have seen those at Wegmans? You have. They're there with the kiwi fruits and the prickly pears, and they come in these little tiny boxes, and they last forever, and you're going to see some samples here. So she's going to tell you about them. I don't want to steal any of your thunder, not that I could. You're done, yeah. But I'm one of the people who's honored to be part of her community science project that Joyce is part of this year, that's supported in part by the National Science Foundation, in part by the Triad Foundation, a local foundation that's been very generous, BTI. And I have a farm, some of you know that, and so we're trialing some of the varieties of husk cherry to try to improve, help them improve the flavor, the yield, and the way the plant grows, the growth habit of the plant. So it's really fun for me to go home and have a little bit of BTI over there in my garden patch. I also want to, so this is a way of, of our integration into the food system. It's really from the discovery. How do we improve this plant? What is it really made of? What are the options? We're not here to become a producer of ground cherries. We're here to discover about them and help through that to maybe help New York farmers, maybe to help consumers have new choices. How many people are here for the first time, by the way, BTI? That is totally cool. I walked in here for the first time in 1988, so that's 30 years ago. I looked really different. I was a California hippie. I had no idea this was the last job interview I did. I had 14 academic interviews, so this is the last one I did. And I realized I was home. So I've never had more than a passing thought of leaving BTI for greener pastures. It's been a wonderful journey for me. And those of you who are here at the first time, your journey starts tonight. And so without any further ado, I'm going to turn the mic over, I believe, to Marissa and then to Nancy.
No, it's okay. With the arrow down, right? Okay. Well, thank you for coming. This is really a pleasure. The last time I spoke to a large group, I was teaching high school. And when I first started teaching at Ithaca High School, we had once a week a group lecture. Picture 125 15-year-olds in one room. It was quite a challenge. It did work. It was a little difficult. They didn't like to sit for a whole hour. Anyhow, thank you, Marissa. She helped me hang the show, and she's been doing a fantastic job of advertising it. Thank you to Stephanie, who invited me to have the show here. We were talking about her trip to the Galapagos, and I said, oh, by the way, do you still have art shows at BTI? And she says, yeah, and I'm in charge of them. I said, aha. Bingo. So I've been associated with BTI actually in many different ways. I had guest speakers at the high school from BTI. We came out, several students and I would come up for field trips to see the research here. I was on a recombinant DNA review committee and reviewed some of your labs. And then, of course, I had the art show a couple years ago. So I feel like I'm part of BTI, having many friends and collaborating back and forth. Okay, so as Dave said, there's a lot of science and related to my artwork. I was a science teacher, and the whole process of science, of discovery, of trial and error, it's integral to all of my photography. The use of light, especially. I mean, if you have a bad lighting situation, your image is not going to look very good. So I've used a lot of the scientific approach that I taught in my photography. So I thank you for bringing that up. I was brought up in a florist family in Schenectady. My grandfather was a florist, my father, my uncles, cousins, my brothers. I didn't realize at that time how central that was going to be to my whole life. GE was in Schenectady, and being a gardener just wasn't really high on the list of social acumen. But it has served me well. I have tons of gardens, and my photography first focused on the plant world. And it has nurtured me in many, many ways. I do flower arranging, I belong to the garden club. So that beginning in a florist family has been phenomenal. And I integrate, a lot of people don't like to talk about plants in the biology classroom. It's not part of the curriculum. I thought it was central to the curriculum. I mean, if you don't have something to eat, you know, you're not going to do very well. So 3 of my brothers went to Cornell studying floriculture. My father wanted me to become a florist. I said, enough is enough in the family. So I studied biology and had that background, but I didn't want to work right away in biology. I always wanted to do something overseas. Remember when John Kennedy said, ask not what your country can do for you, but what you can do for your country? That really hit a chord. Ever since I was a teenager, I'd wanted to go overseas and do something like that. So here was my opportunity. I went to Laos with an organization called International Voluntary Services. It was a pre-Peace Corps. Peace Corps actually modeled itself on IVS. When I was over there, I was a home economist because that's what women do. I also helped them with different, took a lot of photographs and did PR, of teaching villagers what other villagers were doing. So this was networking, early networking. While I was there, I was given a bouquet of lotus. I was pregnant with my daughter in 1968. I was there from 65 to 70, got married over there. My daughter was born over there. So the provincial governor brought this little bouquet of lotus, and I hadn't really seen it before. But that also has been central to my life. It's amazing what that one bouquet did. So came back after 5 years, got a master's, taught biology for 32 years at Ithaca High School, enjoyed it very much, and then retired in 2005 and started a new career. There are classes over at TC3 for adults, and I took a lot of classes in photography and Photoshop, and so that got me very excited about new techniques, new discovery, the science of Photoshop, the science of taking pictures. So, and the rest is history. I've been doing this since about 2007. I was on the art trail. I'm a member of the gallery downtown, and I have shows all over the place because I think, well, I want to share my art, and I think it's pretty. So, let me see if I can go. So in about 1995, a niece of mine was doing masters in elementary education, and she was reading a book about the Vietnamese tradition of taking seeds from a lotus from one village to another, especially during the war, to save their lotus blossoms. And we went down to the farmer's market that day, a sister of mine and I with my niece, and there in the pond were lotus. That little teeny pond in front of the market, there were several lotus, and I asked the person who was in charge, I said, do you ever sell them? And he said, yes. And so I bought 6 plants. And right now, and I planted them just in the side of the pond. I'd had the pond dug out. It was actually pretty dry. The pond is in the back of my yard. It used to be used as a hockey rink by former owners of the house. And so I had it dug into something the size of a hockey rink with a little bit more class. And so I planted these lotus along the side. Basically, it was so dry, I put the lotus down and put a rock on them and said, I hope you're going to live. They did. Right now, there's about 500 lotus in the pond. Vegetative propagation is very rapid. I've had the pond dug out a couple times because of cattail invasion, and even taking out a huge number of lotus, with getting the cattails out, the lotus grew back in about a year. It's really phenomenal. So, oops, hit the wrong thing. Uh-oh. Marissa, I hit the wrong thing. Help. I hit this thing here. Okay, thanks. I'll try to hit the right one. Okay. So when I took these classes at TC3, of course, the first images I was taking were of the lotus. And while I was taking this Photoshop class, I discovered some tools in Photoshop that you could take and extract parts of flowers. I said, this is digital dissection, just like we used to tear flowers apart in the classroom to see their parts. So I said, what can I do with that? So in Photoshop, you can have a canvas, and you can add things to the canvas from your pictures that you've been tearing apart. So think of, this is a full lotus. And then this is a digital presentation of I called them montages. This was my thrill for a long time, is putting parts of flowers together to make a montage, which I thought was very attractive. And you could expand the size of one image versus another. For instance, the pod there is not that big. It's just for artistic interest. So that was the first montage. And then I did several others. Again, it was fun. It was like going back to school and just tearing flowers apart and putting them back together into something I thought was pleasant. If you look at the center of the lotus is the to-be pod. And you probably have seen these pods in different floral shops. The pod is actually the fruit of the lotus. That's where the seeds develop in that pod. And the seeds are actually edible early on. And you can eat the pod when it's green, not when it's brown. By the time it's brown, it's way past anything edible. So, this is restudying my anatomy of flowers. So these are some of my favorite montages. Then, because there's a lot of lotus and Buddhist art, I thought, well, maybe I can combine the two. And I put a lotus overlay over a Buddha image and made it translucent. I also did it over the clock tower, just for fun. A friend of mine who's here tonight suggested I take this class in New York City of macro photography. It was a weekend, I think it was Easter weekend several years ago. The group, it was in somebody's apartment. We went to the floral market in New York City, which was a thrill to me because my brothers and father and grandfather being florists, they got a lot of their flowers from the flower market in the city. So it was a thrill to go to the flower market. We picked up flowers and took them back. And she spent the whole time doing macro photography. That sort of changed my emphasis. Before I was doing all these montages, then this macro photography really made sense. I use a macro lens. I have the Canon camera, the macro lens, and definitely using a tripod. I tried to cheat and not use a tripod. I said, I can do this. It's never quite the same. So clarity, which is scientific, is important in macro. It shows up more with macro than it does with just a scene. A scene you can get away with a lot. You do macro and you know if you've moved that camera. So I take macro images in the garden, and I have a greenhouse because I grew up in a floral family, and I set up a black cloth on a rack and put the flowers in front of it, staging them, just like a portrait. And I usually wait for afternoon sun to come through the greenhouse because it's a soft sun. Doing something in the middle of the day, it washes out your flowers. So afternoon sun is soft, it enhances the flowers. All of those macro images out there, most of them are from my garden, and they were all taken in the greenhouse, waiting for the sun to go down. I don't like using artificial light. I think that it doesn't have the same warmth for the flowers. So you've seen a lot of these out there. This is a dahlia in the garden. Again, afternoon sun, very soft. This is, Marissa used this for the ad, and it was one of my favorite flowers. It was a lotus in the pond. It was just a perfect day. A lot of the times The lotus flower could get washed out if the sun is too bright, but this was perfect. So inside, again, these are out in the hallway. The bleeding heart, daylily, and bridezilla. I love that name. The name of the dahlia is called bridezilla. Bee balm, another dahlia. Sometimes I add water to cheat because it looks nice, but often if it's been raining, I just bring the flowers in after the rain and it's a natural moisture. Now these were the ones I did in New York City. She used a white background. I happen to prefer the black. Somebody the other day said, why don't you use blues or greens or pinks? So, because I like black, that's why. I mean, obviously I like black. So again, the anatomy of the flower, these are, at least the one on the right is out in the main building there, and you can see the clarity of the to-be pod. Green, the green gladiolus I thought was quite striking, and the orange. This was an interesting, a friend gave me these flowers, and she said they weren't at all like what she had the year before. There was some kind of a spontaneous mutation. I have no idea. She didn't know. She said, but they were not that color last year. And of course, she hasn't gotten them back either. I have no idea. If somebody knows, let me know. More of the lotus. They're very aromatic. A lot of, in Egypt, they have a blue lotus, and that's a source of a lot of perfumes. In fact, they use that as a base for many of the perfumes in Europe, I was told. So it's a wonderful smooth scent as you walk next to the pond. We've got the poppy on the left was from New York City, and again, that golden dahlia. More lotus. Right now, the first lotus leaves are up. The first leaves look like a water lily. They're flat on the surface, and so that tells me that it's going to be another successful year. When we have some of these horrible winters, I thought they're not gonna last. But the first leaves are up, and the second leaf, the one that looks like an umbrella, will be coming up early or about mid-June, and then the flowers will be blooming in July. The first flower, it's like having a baby. I mean, it's just, whoa, it worked again. I entertained myself. So then I got into making floral bouquets in front of this black cloth. And here you can see all the different stages of the lotus. You've got the bud, the early pod, the later dry pod, and then the very flush flower. I can have, these are all printed by a company. It's called Canvas on Demand Pro. I send digital images off, tell them what size I want, and I get the canvases back. The last show I had here was of flowers that I printed, I matted, I framed. I like this better. I send it off, it comes back, and I get excited. Oh, isn't that beautiful? Anyhow, you can order groupings, and this grouping happens to be at Mansour Jewelers right now. So my other passion, well, I have a lot of them. I take zoo animals and raptors and other things, but one of my other interests is the cement sculptures in the Arboretum. And they were produced in the 60s by a graduating class from the architecture school. Jack Squires was their professor, and he had them make these sculptures. They wanted to put them on the main campus. The administration of Cornell did not want them on the main campus. So where did they put them? Where they are now. But what was there then was a field of grasses and cow stuff. It wasn't very pleasant, I guess. It was very overgrown. So eventually, when the Arboretum was recognized more as an Arboretum, they started cleaning it up. And just the last couple of years, they really cleaned it up and gave it signage. So it's gorgeous. Some of my interest in a lot of this is because I really liked Picasso's art and Picasso's cubism. And so these reminded me of his art, and what I did with it definitely reminded me of his art. True Picasso experts might not agree, but to me, I'm doing somewhat cubism. Okay, this is an alumni news, I think it was 1961. And it was on the cover. Somehow I found this and scanned the image. They called this the teaching rings for architects. And the article said, Squires said, doing sculptures was a good teaching opportunity for architects. They could start with a sculpture and then expand from there. So this shows the students working. A lot of the students had to pay for part of the art themselves. I guess there wasn't a very good budget at the time. So you can see the conditions when they finished these sculptures. It was weedy and there's cow stuff and everything else, and not very well taken care of. I have a lot of former students who have said that they used to climb on these when they were growing up. Any kids in the neighborhood would go up there and climb on them. Well, now these are some of my images. So I start with, again, images of the sculptures. Try to get the right time of the day, the right light on it. If you get it too bright, it just doesn't look very good. I love some of the shapes. These are the ones that look like teething rings. These students had great imaginations, and I can imagine how difficult it was to construct something of cement that high. Okay, so then I used the technique in Photoshop that I'd used originally for the flowers, that tool that you can extract parts of something. And so I made a digital black canvas on Photoshop, and I had some of my pictures, and some of them are more torn apart than others, but I could take stuff from one picture and put it on the canvas to make a piece of art. This is out in the main building. It's fun. You never know with this tool exactly what you're going to come up with. If it doesn't look good, you throw it out. Digital photography is cheap. Easy to throw away. I incorporated some trees with some of them. This is more montage, where I'm combining several different images. This is probably the most montage with pieces from all of the different sculptures. I'm within my 20 minutes, right? It's been a fun way to spend retirement. It's been educational, nurturing, a challenge. It's certainly, every day I can think of what can I take a picture of today and what can my next project be. So it's been fun. Thank you for coming.
How do you use depth of field here? Try to maximize depth of field or minimize it?
Well, if it's outside, you don't want a lot of depth of field because you want the blurring in the background. Inside, with a macro lens, you want the depth of field to see the whole flower. So putting it on a tripod, you can get away with low light and get decent depth of field. That's why they almost look like sculptures sometimes, because I have gotten the depth of field.
Another question. If there might be a little bit of blemish on the petals, or do you touch it up a little bit? Or are they
You know, I've tried, there's this thing called a clone stamp. It never looks the same in my experience. Maybe somebody else can do it better, but it always looks like you're fooling around there. I try to get another flower. You know, there's enough dahlias in the garden at the end of the season, and if one didn't work, you toss it.
Thank you so much, Nancy.
You're welcome. Thank you.
So up next, we will have BTI Associate Professor Joyce Van Eck.
Okay, all right. Thank you, Marissa. It really is great to see such a big group in here, and thanks for staying for my talk as well. So I'm going to turn the lights down a bit because I think the colors show up better with a little bit lower lighting. Okay, just don't fall asleep on me. Okay, all right, the remote. All right, so this slide contains images of some plant species that belong to the Solanaceae family. Yes, plants have families too. The Solanaceae family is a very diverse group. It contains about 3,000 different species. These are some, oops, nothing but turn them away. Oh, there it is, okay. These are some native potatoes from the Andes. These are tomatoes. I mean, the amount of diversity in tomatoes is just amazing, isn't it? This is a petunia, pepper, some eggplants. This is something called tree tomato, and it's not that one you see in magazines that they put on a stalk. This is actually something called, it grows on a tree. Some other tomatoes here. And I've been working with tomatoes since I was an undergraduate, and my group continues to spend most of its time working on tomato projects. But most recently, we started working with this Solanaceae family member, Physalis, specifically this little round yellow fruited one. And Esperanza Shenstone, who's a member of my group, is going to pass out some baskets of the fruit. Give them a try. Take them out of the husk. That's not edible. See what you think. You don't have to try them, but you're welcome to give them a try. Okay. All right. So how did we get started working with the Physalis? Well, as I mentioned, I've been working with tomatoes quite a bit, and we had a project with tomato that was funded by the National Science Foundation, or NSF. And the goal of that project was to look at the genes involved in plant habit and fruit characteristics. So if you've grown tomatoes, you've got those ones that are really long and wild. They're called indeterminate, and often the cherry tomatoes are like that. And then you have more compact or determinate type tomatoes. So we wanted to understand, what are the genes involved and how does it function and what makes the difference? And the other characteristic, as I mentioned, were fruits, and we're looking at fruit size and mainly in weight and in the size. So we accomplished a lot. We learned a lot. And then the next phase, what we wanted to do was to see, can we take what we've learned in tomato and apply that to a more distantly related relative in the Solanaceae? So what I have here is called a phylogenetic tree, and it shows the relationship of some of the Solanaceae family members. So when we were looking at this and trying to decide what distant relative to use, we decided to work with the Physalis. And the Physalis that you're probably most familiar with are the tomatillo, or Physalis ixocarpa. But we chose to work with Physalis prenosa, ground cherry, which is being passed around, and Physalis peruviana, or goldenberry. And besides the biology of what would be interesting to see what worked in a more distantly related species, we also felt, my collaborators, we felt that there could be some potential for these to be a specialty fruit crop grown in New York State and possibly beyond. So the origins of the ground cherry are Mexico, Central America, and goldenberry is in the Andes region. So you can see they have this little fruit, this husk around the fruit, and this is the inside of a fruit. Kind of looks like a cherry tomato, but it tastes very different. Do you think so, for those of you who tasted them? What do you think? Good, that's what I like to hear. Okay. So they're very sweet and very juicy. Okay. So they've also been around for a long time. There was a report last year about fossils that were found in Patagonia, Argentina, and they dated them back to 52 million years ago. They originally thought to be 9 to 11 million years old. And I'll talk a little bit more about this husk or this inflated calyx, but it shows a very ancient history. I just think these images are so cool. And you can see there's kind of the remnants of a fruit in there as well. If you want to, read more about it, there's this URL here, or if you just do a Google search for Physalis fossils, you'll turn it up. So some other characteristics of ground cherry and goldenberry are that they're an underutilized crop in the US, and we're hoping to possibly change that. It's more popular in countries such as China, the Netherlands, and Colombia. And actually, Colombia is the main exporter of goldenberry. And a friend of mine told me that I had never seen them in the supermarket. And a friend of mine told me he saw them in Tops. And I said, really? So the first chance I had, I went to Tops. I was looking in with the tomatoes. I couldn't find them. And I thought, what is he talking about? I don't see them. But as I passed the raspberries and strawberries and blueberry section, it really stood out because it was this bright yellow. So I paid $5 for that little clamshell. But I thought, okay, this is my work. I took it home. I washed them. I tasted them. They were horrible. They were like the worst tasting. And I thought, we have much better tasting germplasm in our group. But yeah, I was really, really disappointed. Anyway, they've got a good nutritional content of vitamin C, beta carotene. They have some anti-inflammatory and antioxidant components. I'm seeing more reports of medicinal properties related to these fruit. And they also contain compounds for plant protection, so to protect them against pests and diseases. So this shows the plant habit of both of these, and similar to tomato, they just keep growing, and I have a small garden, and I didn't stake them in my garden, they just took over. This shows the ground cherry and the goldenberry. And one of the characteristics or traits that we want to improve is to make them more compact, make them more manageable to grow. So what we've been doing is looking through what's called germplasm. We have a collection from different countries, and those that are in these yellow boxes are the newest germplasm in our collection. But for those that we've had the longest time, we are seeing some differences in the fruit we do see differences in fruit between the Prenosa and Peruviana, but we're also seeing it from the different places where seeds have been collected and they've been grown. So the one from India, actually has kind of a coconutty type flavor to it. If we had some, I would have brought those as well, but we didn't. We only had these that are from the Netherlands, the ones that I passed around. And the South Africa one has kind of a citrusy orange flavor to it. And one of the Poland ones, which is called Aunt Molly's, which is, if you might have heard of that, it has a pineapple flavor to it. So it's been really fun to taste these different fruits as well. We see differences in the flowers, and I included some flower pictures as I thought Nancy might be inspired to do some artistic photography of chrysalis flowers. This is goldenberry, and you can see that it's got 5 petals, 1, 2, 3, 4, 5. And these centers here, there's a little bit frilly, and it's got these dark lines between the petals. But in the ground cherry, it's a little bit smoother, these brown areas, and you don't see those lines between the petals. But we're seeing some differences there as well. Now, what's with the husk? So the way the husk forms is that this is a young flower bud, and this region is called the sepals. And as that flower develops, those sepals elongate, and they eventually encapsulate the fruit. So as you can imagine, that husk prevents it from being eaten by insects, but also being attacked by diseases. But the other added benefit is that it gives it a really long shelf life. And I have an example. I gave a talk about 2 years ago at the University of California, Davis, and a graduate student brought me a couple of bags of fruit from her gardens. So I brought them back, we tasted them in the lab, I put them on the shelf, and I said, we'll get some seeds out of those. About 4 months later, I realized they were still on the shelf in my office. And I thought, oh, afraid to open that bag, it's just gonna be mold and fungus, all sorts of things growing in there, and they were perfect. So I took them out of the husk and I tasted it, and aside from being a little dehydrated, they were still juicy and they tasted really good. So that husk really gives them a longer shelf life as well. And the goldenberry and the ground cherry are eaten as a fresh fruit, but also they're used for jams and juices as well. I mentioned the plant growth habit is one of the things we want to look at and try to improve. The other thing is that ground cherry, hence the name ground cherry, has a problem with fruit drop. So it just drops even before it's ripened. You have to gather it up from the ground. And it also, not only being a difficulty with harvesting, it also imposes a food safety issue when you're trying to gather up fruit from the ground. So this is one of the main traits that we want to look at for ground cherry. And then somebody asked me, well, then what would you call it? And I don't know, because it won't be dropping to the ground, but we'll deal with that later. So what we're doing is we're evaluating the germplasm that I mentioned to look for lines that possibly don't drop fruit as much. The goldenberry does not drop its fruit. It hangs on. Even when it's ripened, it's still on the plant. So we want to do some crosses, so doing pollinations, taking pollen from Peruviana and Prenosa and in the reverse to see if we could fix that, that drop problem. So the drop is because of something called a joint or an abscission zone layer, which I will show you here. So this is, these are some plants in the greenhouse, and they're small plants, and we have to put trays under them to collect the fruit. But when the plants get really large, we just can't contain the fruit drop after a while. And the reason this happens, I'm just going to zoom in on the fruit. So this is the fruit. This is called the petal cell. And if you see up in this region here, there's that joint or abscission zone layer, and that's what causes the fruit drop as the fruit matures. So we're hoping that we can somehow understand the biology and make the improvement on that joint. Get rid of it. Okay. So what I've told you about now has all been supported through our National Science Foundation funding. But what I really wanted to do was I wanted to get this material into the hands of farmers and the home gardeners, because I wanted to hear from them what improvements needed to be made. And we know there's plant growth habit and there's harvest issues, but what else? But unfortunately, there wasn't enough funding in our NSF project to do this. So I was pretty disappointed. But I had a couple of people at BTI encouraged me to submit a proposal to the Triad Foundation, and David mentioned that earlier. So the Triad Foundation is a locally based foundation. It has a number of initiatives, and one of the things it does is it gives money to BTI, and the researchers on a competitive basis can use these funds to fill in the gaps where other funding agencies don't. So I submitted the proposal, and I called it the promotion of 2 physalis species as new specialty food crops. I got the funding, and I still get goosebumps. And so I'm so thrilled to be able to do this, to have this work. And there are 3 aims. So the first aim, as I mentioned, is to get the material in the hands of farmers. And the way we did this was we worked through Cornell Cooperative Extension Agents, and we have 12 different farms, and they're indicated here by these little red balloons on a Google map. We've given them seeds of 3 different types of goldenberry and ground cherry, given them surveys to get feedback, to hear from them about what they want. There'll be visits to farms, Esperanza, and our intern are going to be visiting farms this summer. And then in September, we're going to have a harvest event where we're going to have some taste testing and sharing experiences with growing them, and have some foods prepared with the physalis. Aim 2 was to engage the consumers, to get this material in the hands of home gardeners, and we're calling it a citizen science project. It was great. I sent an email out to the BTI listserv. I said, anybody interested? And I got an overwhelmingly amazing response. We've got 23 participants, and as David mentioned, he's participating. Jay is as well. I don't know if anybody else is here that is, but anyway, it's been a lot of fun. Also in Illinois, Iowa, and New Jersey, so through family members, and also my giving talks in different places, people have asked me to give them seeds. Community gardens in the Ithaca Children's Garden and Keegan Nature Center are going to make it part of their summer camp. So where the children will be taking different observations and making measurements, and Esperanza is going to be visiting to give them a presentation. To help facilitate communication, we have a Facebook page. There's a screenshot of the Facebook page here, a web page and a blog. And part of the blog, Esperanza occasionally puts out something called a biology minute. And she, talks about the husk, fruit ripening, and other characteristics that would be of interest. And the participants in this will also be part of this harvest event in September. And this just shows a screenshot of some of the blogs that Esperanza has posted. And this is our webpage. If you're interested in taking a look, you don't have to remember this URL. Just go to BTI, to our website, and go under the research menu, and you'll find the Physalis project. Third aim is to involve an intern and have a student who's interested in plant breeding. I want this intern to visit the farms to learn the importance of listening to their stakeholders and gearing their projects towards what the needs are. We're also going to give them some hands-on experience with doing crosses and also evaluating populations, which is a big part of the breeding program. So basically what we're doing is we're giving them experience with approaches that help support crop improvement efforts. Okay, and with that, I have a number of people to thank. The members of my group, Nathan, Esperanza, and Carrie. Gina Hall helps me put my grants together. Our advancement team, our group from communications and development, and Delaney from our outreach. Kelly and Aurora helped plant seeds and transplant plants, which is great. Our greenhouse group, Brian, Brittany, and Jay, thank you for all your patience with all those fruit that dropped to the ground, and we can't get to them too soon. And my collaborators at Cold Spring Harbor and Johns Hopkins, and especially to the people who give us the funding, because otherwise we couldn't do this work. And thank you.
But we do have a little bit of time for one or two questions.
So, as you're working to introduce new crops in the area and stuff that may not as familiar to the local community, do you also engage local restaurants or chefs or anyone in the culinary community to see about and how to integrate it into the diet?
That will be the second phase, which the Triad Foundation, I'll go in again and see if we want to do a second phase and do exactly that. That's an excellent point. That would be part of that education engagement program as well. Yep. So if you have any connections.
Actually, I do.
Oh, great. Well, we need to talk. Okay, great. Good. Anyone else? Yes? Ah. And it gets in the fruit. Up until now, we've grown them mainly in the greenhouses, and this is our first year of this big trial. So that is the type of feedback I'm loving. To hear that you have that problem. I did grow them in my gardens, but I never got fruit because the chipmunks got to them before I could. Yeah, I was wondering why I wasn't getting any fruit. And I'd pick up the plant and go, well, I have husks and the husks are forming. Why don't I have fruit? And then I noticed on my compost bin, on the lid, they were empty husks. And I saw a chipmunk just go over and I was opening it. So this is again, I gave up last year. I'm trying again this year, but it's good to know. We'll have to talk a little bit more about that. You can describe. Yeah. Yeah, that would be a real turn off. Yeah. Yeah, but that's good to know. That's again, that's one of the reasons why we're doing these big trials, is to find out what the other issues are that we don't know about from growing them in the greenhouse or just in our our gardens on a small scale. Thank you.
How
how do these compare in cold hardiness? Would there be an option for high tunnel kind of production off season?
We are, so there's a group in Geneva who is doing high tunnel trials for us as well. So that's, and I'm wondering if maybe possibly for the origins of some of these in the Andes, there might be some, you know, more extended season to them. But we'll know about this high tunnel. That's the answer point. Yep.
Are they grown in greenhouses, for instance, in the Netherlands?
Probably. I don't know. It's a great question because I do a lot of production in greenhouses. So I'm in the Netherlands. So I'm imagining they do. I should look into that and find out. But I wouldn't be surprised if they do. They grow really well. For those, I don't know if anybody signed up for the greenhouse tour. I hope you did. Oh yeah, one of the things I forgot to mention. If anybody wants plants, we have some extra plants. Oh, okay. Do we have enough Esperanza? All right. So, so if you're on the greenhouse tour, we'll hand out some plants to you. If you're not on the greenhouse tour, we'll bring them out for you. Did the greenhouse tours get filled up? Or?
Yeah. So because it's timing, we are going to combine greenhouse tours this evening.
Okay. Okay.
I believe we still have a couple species left, so if you'd like to sign up, Allie with the clipboard in the back is available. So she will be congregating people who are interested in going on the green for outside these tours.
But if you don't get to go, just let me know, and if we have the plants left, I'll give you one. And we'll give them, you know, whatever we have, you know, from Poland or whatever country. So, okay. Thank you. This was a lot of fun. Thanks.
I believe the GABA is still, there's still some food outside, there's still wine, so if you'd like to continue to mingle, I encourage you to do so. And